So welcome everyone. It's a pleasure to be talking to you today. Thank you for attending. I'm Sam. I'm a postdoc in the Pathogen Informatics and Modeling group at EMBL-EBI. And today I'm going to be giving an overview, an introduction to pangenome graphs and why I believe they're a new paradigm in comparative genomics. Just to give an overview of what I'll be talking about. For some of you, this might be sort of rehashing previous sessions. There might be some of you that just need a bit of refreshing, and maybe some of you weren't in the previous sessions, but I'll go through what a pangenome is and why they're interesting and why they're important. I'll then introduce pangenome graphs and why we would want to represent pangenomes to the graph. Then I'll talk about different types of pangenome graphs and their pros and cons. And then I'll give a case study for an application of pangenome graphs, which is an application that I developed, ggCaller, also known as Graph Gene Caller, very simply, which is a bacterial gene annotation tool. So what is a pangenome? This is the observation that the collective genomic diversity in a population is much larger than that found in a single genome. So that's quite wordy. What do I actually mean by that? Let's look at a cartoon. So if I were to take 3 different genomes of 3 different individuals, and in bacteria, which is the sort of the taxa that I work on a lot, my main focus, we tend to look at the genic content of genomes because bacterial genomes are gene rich. And by looking at genes, we are sort of capturing most of the genomic diversity in a population. If we call the genes represented by these colored blocks on a genome, and then compare them between these different genomes by sequence identity measures, or maybe even structural identity measures, depends on sort of the method you're using. If we were to then look at the sharing of genes across these genomes, we tend to observe 3 distinct compartments of genes based on their frequency. So rare genes that are only found in one individual, intermediate frequency genes that are found shared between multiple individuals, but not all individuals, and then core genes that are found to be shared between all individuals in the population. But why do we care about this? Why do we care about pangenomes? So particularly in bacterial analysis, core genes allow us to analyze the evolution and genomic epidemiology of a population. So by taking core genes and aligning them and comparing them and seeing what differences are found between these different genomes, so by aligning these genes here, we can then construct phylogenetic trees, look for potential outbreaks, so disease-causing members of a bacterial species, differentiating them from other circulating individuals that might not be causing disease but are just present and carried in the population. Whereas looking at accessory genes gives us an idea of the within population diversity of a species or genus or whatever taxonomic level you're looking at. And this is particularly useful in conducting genotype-phenotype association. So many of you may have heard of genome-wide association studies in say, humans looking for statistical associations between genetic variants and a phenotype. You could do the same thing in microbial data. So in bacteria, we tend to look at, well, a lot of interest is garnered around looking for the genomic and causal genes involved in drug resistance or antibiotic resistance. And using such approaches, we can look for the causal genes that are giving rise to drug resistance and differentiating them from variants that don't give rise to that variant, resistance. Now, why would we want to represent a pangenome as a graph? So a pangenome is a collection of all of the genomic diversity in the population, but there's multiple different ways we can represent this. Conventionally, before the advent of pangenome graphs, we would tend to represent collections of genomic diversity just as collections of linear sequences, as you would do looking at a normal genome. So a genome can be represented as a one-dimensional string of A, C, Gs, and Ts. And they contain redundant information in the form of shared gene content. So if we were to look at 3 different genomes, A, B, and C, and align them based on their linear sequences, it's very easy to identify small variants, so SNPs, single nucleotide polymorphisms, using this type of approach. And this is sort of why SNP-based variant calling is so popular, because it's easy to do when representing genomes as linear strings. It's much more difficult to look for structural variants, so translocations or deletions, which shift the order around the gene of the genome and make it much more difficult to align these three different linear sequences in the order that they're found. So pangenome graphs allow us to get around this issue of limited alignment of these large-scale structure variants. So we can take the three genomes shown above and collapse similar sequences down into nodes and represent variation between them as edges. And this is an example now of a pangenome graph. What we can see from this pangenome graph is that it's lossless, which means that we don't lose any information present when representing genomes in a linear format when representing them as a pangenome graph. Because we can follow the colors of the arrows through this graph and recapitulate the original sequences that we used to build the graph in the first place. What you might also note is that this format is much more compact. We're storing the same amount of information in a much smaller data structure, which means that it's easier to load into memory, manipulate. Also, it's a lot easier to identify variants in this case. So we can look for these structures called bubbles induced by, say, SNPs here. So we can see a bubble caused by just a single nucleotide variant. But we can also much more easily identify deletions here or translocations based on following paths through the graph. So overall, just to summarize all that up, why would you use a pangenome graph? In essence, it's easier to identify large-scale variation and small-scale variation. You can also identify core and accessory content based on the frequency in which these nodes are found and how frequent the variants are found as well. And they're also much more compact. So sort of a winning formula for conducting comparative genomics. But it's not that simple. Just wanting to say I want to build a pangenome graph of my genomes. There's multiple different types of pangenome graph, and each of them have their own caveats and associated with them. And there's two major subtypes of pangenome graph that I'll talk about. The first are alignment graphs. So alignment graphs are built from multiple sequence alignments, so taking your linear sequences, aligning them together, and then looking for how the variants are shared or different across your multiple sequences, or from variant call files from VCFs. They can be built from undiverse or diverse populations. So the alignment graphs have been used to build multi-species pangenome graphs. Some of the most impressive ones I've seen build pangenome graphs from multiple diversified species, so whole genome alignments of apes, for example. They're highly flexible and can capture small and large-scale variation. The main caveat, though, with alignment graphs is that they scale poorly. So they're slow to build and to align to because alignment is inherently slow. In comparison, on the flip side, you can also build pangenome graphs via alignment-free methods. And these are built de novo rather than conducting alignment to build the graph in the first place. They use pattern matching or k-mers in this case. And I'll go into a bit more detail about what k-mers are. All a k-mer is a nucleotide sequence of length k. So you can have a 2-mer, which is 2 nucleotides long. You can have a 3-mer, which is 3 nucleotides long. And you can build pangenome graphs using k-mer matching, which makes it very, very quick. You're not conducting base level alignment. You're just matching identical seq patterns across a data set. The main issues with using alignment-free methods are that they become inaccurate in highly diverse populations. Because k-mer matching becomes less likely, you're less likely to match k-mers when your two genomes you're trying to match have more diversity between them. They have fewer shared k-mers. And so, in my opinion, you should only be really using alignment-free methods when you're looking at a single species population. They're also more or less flexible than alignment graphs. You have to normally pick this k parameter, which depending on which k parameter you pick, restricts the variation you can represent. So too small a k means that you miss your ability to look for large-scale variations, so these structural variants such as translocations and deletions, whereas a too large a k means that you're unable to resolve very small variation, such as SNPs. The main bonus for using alignment-free methods, though, is that it's highly scalable and it's fast to build these graphs and to align them. And in some cases, I've seen you can build graphs from hundreds of thousands of genomes of the same species. So just going into a bit more detail now about what the different types of alignment graphs are, because even within alignment graphs and alignment-free graphs, there are variants that have their own caveats again. So the first recorded that I could find mention of Pangenome graphs was in a paper by Hein in 1989, which was used, represented each base in an alignment as a single node in the graph. And it was deemed as a scalable method for aligning tens of kilobase length sequences. So in this case, it was used for generating phylogenetic trees of genes. We now have methods that scale up to genome-sized alignments, and a good example of this is a cactus graph. This was published a few decades after the original alignment graph paper by Hein. It encodes nested variation in these sorts of buds, like a cactus-like structure, hence the name. And it's used to generate large whole genome alignments. And if you remember a few slides ago when I mentioned these really impressive alignment graphs that are built from can be built from whole genome alignments of apes, this was the tool that was used to do this. And just to sort of walk through how cactus graphs work. And represent variation. Each thread, so this green and blue line running through this graph represents a genome, and each node represents a hierarchical variant, a large or small variant. So this node A1 represents maybe a large translocation. Within that large translocation, we have multiple other potential variation. So this might be a smaller translocation nested. So B1, node B1 might be a smaller scale variant nested within this node. And then nodes C1, C2, and C3 may well represent an even smaller variant, so maybe a SNP. So think about this large node A1 as maybe a big translocation. This node B1 as an indel, so a smaller variant, and then C1, C2, and C3 might be SNPs. And so you can represent, by following the thread through each of these nodes, you can then determine which variants are present in each of the genomes and which ones are shared. You can see that, say, node C1 is only found in the green genome, whereas node C2, there's potentially a SNP, is shared between the green and the blue genome. The main issue with using cactus graphs are there's two main issues. They're slow to build, and also they're very difficult to interpret in this way. You need to really think about what the nodes are representing in this case. An improvement upon, I would say, cactus graphs are variation graphs. So these represent all scale, a hierarchical scale variation, but at a sequence level, and can be used to generate whole genome alignments. They're much more easily visualized than cactus graphs. And there's actually a toolkit called the VG toolkit, the variation graph toolkit, which can be used to convert cactus graphs into this more human readable format. A further variation on variation graphs is this are graphs represented by this tool called minigraph. Rather than representing all scale variation, so SNPs and large scale variants, minigraph conducts only structural alignment and is reference based. So given a given reference sequence, minigraph progressively aligns a number of different genomes to the reference and only adds in variation which is larger than 50 base pairs in length, for example. Because it's reference based, it can actually, the graph you get out may well be biased by the reference chosen, and depending on what reference sequence you pick, can actually mean you get different graphs out at the end, which isn't the case for, say, using a variation graph or a cactus graph. A further variation on these variation graphs are represented by a pan graph, which is another tool which conducts base level alignment like, say, a variation graph, but represents variation in terms of homologous blocks. So within each homologous block, you can conduct base level alignment, so you can actually call SNPs within each of these homologous blocks. But like a mini graph, pan graph represents structural variations as breakages in this graph. So you can actually very easily pull out small scale variants such as SNPs by looking for variants within these homologous blocks, but also look for these large scale variants which are represented by these breaks in the pan graph itself. And this has been shown to work really nicely for doing bacterial pangenome analysis. I just wanted to give a brief nod to gene graphs. These don't really fall into either of the alignment or alignment-free graphs. I believe Neil would have mentioned this in the previous webinar on analysis using Panaroo and Rory. Gene graphs, in the same way as other pangenome graphs, simplify and compact down variation from a linear sequences to a graph-based format. But rather than working on a nucleotide level, so merging nodes based on the presence or absence of A, C, Gs, and Ts, these gene graphs merge whole genes together into single nodes. So imagine that we have called all the genes in a linear genome, in a yellow genome, a blue genome, and a green genome. Tools such as Panaroo build gene graphs to improve gene prediction correction. So this would be the corresponding gene graph if we were to call all the genes and merge them into a pangenome gene graph here. And I'll go into a bit more detail later on. I've got another slide on Panaroo later on. What Panaroo allows you to do is to identify very easily core genes and accessory genes, which, if you remember back to a few slides ago, it's important that we're able to identify what's core and what's an accessory sequence. There's also some tricks that Panaroo uses to enable improved annotation, so refinding genes. If a gene is missing in, say, the green genome, but present in the yellow and the blue genome, it can use that information to actually search for that gene in the green genome and refind it if it was misannotated, for example. And there's some other tricks that it uses as well that I'll go into detail in a later slide. So that's alignment graphs covered. Now to discuss briefly some alignment-free graph formats. The crux of alignment-free graph building are De Bruijn graphs. These break up genomes, also denoted as colors, into overlapping k-mers. And these, as I said before, k-mers are nucleotide sequences of length k. The idea behind De Bruijn graphs are that you just need to match identical k-mers across the genomes you're analyzing rather than conducting slower alignment. So if you're building a De Bruijn graph, you may well call, starting with a k-mer size of 6, identify the first k-mer that's present in a genome, and then look for the genomes in which this k-mer is found. So we can see that it's found in genomes A, B, and C from the colors of the k-mer. To then generate the next k-mer, all you need to do is drop the first base, so drop this A here, and then get the next base that's present in one of the genome sequences. So in this case, it would be a T, and you just do that moving forward and slide forward in a window of size 6, whilst also taking into account which genomes each k-mer is found in. These are the colors. The genomes denote the colors denote which genomes k-mers are found in. And you move through each genome, identifying which k-mer is found, just matching it based on its presence or absence in other genomes. When you hit a variant, this induces a bubble of length k, because as you imagine, if you slide through a window which contains a variant which is found in one genome but not another, that variant is going to move through your k-mers stepwise until it's pushed out the other side. What you might notice is that these De Bruijn graphs contain a lot of redundant information in terms of the degree of overlap between neighboring k-mers. So for example, we can see that this first k-mer here has an overlap of size k-1 with the next k-mer along with it. This k-mer has an overlap of size k-1 with the next k-mer, and so on and so forth. And this is very wasteful. You're sort of repeating the same information over and over again. And this means that De Bruijn graphs, when uncompacted, take up a lot of space either on disk or in memory. And so a variation on these original De Bruijn graphs are compacted De Bruijn graphs, where these long stretches of k-mers which have k-1 overlap are merged into unitigs. You can see here we have the same information as before. We can see which genomes each unitig is found in. So A, B, and C here. This unitig is only found in genome A or in genome B and C. This is much more compact, easier to store, and more efficient to work with. And this, rather than using uncompacted De Bruijn graphs, I'd always suggest using compacted De Bruijn graphs. There's no real reason why you wouldn't want to do that. So just to give an overview of using different pangenome graphs, there's a number of different pangenome graph building tools available. How do you choose which one you want to use? And just some pointers that I could, so hopefully will guide your choice. The things to consider are scalability. If you have a very large data set, alignment is probably not going to really work. It's going to be very slow. Alignment free is probably better. On the flip side of that, alignment is very good for capturing high and low diversity populations. Alignment free is only suitable on low diversity populations, and this stems down to the issue of k-mer mismatching. If you have a too diverse population, you'll get a lot of mismatching k-mers. You won't actually be able to see and match anything across your different genomes, and you have a very disjoint graph. You can also have to think about usability. Do you need your graph to be human interpretable? So, for example, cactus graphs are quite difficult to read and understand. Also, are there tools available to help you work with the graph? The VG Toolkit is super useful for sort of manipulating any kind of variation graph that you could possibly need, and it's why it's so popular as a tool to work with pangenome graphs. So now I'm going to move on to a case study for using pangenome graphs in bioinformatics analysis. I'm going to talk about ggcaller, which is a tool that I developed during my PhD. So just to give an introduction to how conventional gene prediction is conducted and why you might want to do this in a pangenome graph. When you sequence bacterial isolates and want to conduct pangenome analysis, what you tend to do is sequence your isolates, assemble them individually using tools such as spades, identify the genes that are found on each of the genomes. And as I said before, in bacterial analysis, we're really interested in the gene content of a pangenome because genes are or bacterial genomes are gene rich, and capturing the genic content gives you a very good idea of what the population diversity is. So you call genes individually in each genome using tools such as Proper or Bacter, and then you conduct gene clustering, which would have been mentioned by Neil in the previous webinar, using tools such as Rory or Panaroo, as I mentioned before. And from this, you can then generate a gene presence absence matrix. So determining which genomes by row have which genes by column. And by generating this, you can identify what are your core genes, what are your accessory genes, and then conduct your downstream analysis, whether that's building a phylogenetic tree or conducting genotype-phenotype association. The motivation behind ggcaller was to skip a lot of these processes, and on the top row, conducting graph assembly, either building from reads or from assemblies, and building a De Bruijn graph, and then using the frequency level information, so looking for call nodes or accessory nodes in the pangenome graph, using that information to call genes on a graph itself rather than individual genomes, and then being able to go from a graph to generating a gene presence absence matrix. And I'll talk a bit more about the motivations as to why and what some of the benefits we see when we use a pangenome graph versus annotating genes individually on individual genomes. So the first thing is that related genomes will contain redundant genes, and so this will take quite a lot of time, although you can parallelize it. So imagine you're moving through each genome one at a time, calling genes individually on each genome, whereas using a graph, you can traverse the graph, calling genes in the population all at once, and hopefully speed up this process. And also sharing information across genomes, you can improve consistency in gene calling between related genes as well. So the way in which ggcaller works is that starting from assemblies, the De Bruijn graph or a compacted De Bruijn graph is built using Bifrost. This is a highly scalable compacted De Bruijn graph builder, which I've cited down the bottom if you're interested. There's a really nice API to work with as well. I'd recommend if you wanted to develop any tools for pangenome graphs, this is definitely a great tool to use. So once you've built your graph, ggcaller then identifies all of the stop codons across the entire population, across the entire pangenome graph. And the reason why ggcaller looks for stop codons is that bacterial genes don't contain introns, and so therefore a bacterial gene will only ever be found between two stop codons. It can't traverse two stop codons. So it always begin somewhere within these two stop codons and end at one of the two stop codons. Starting from an individual source node, ggcaller then traverses outwardly from a stop codon to pair up all stop codons in the same reading frame. So therefore, once we've paired up these stop codons, we can be confident that if there is a gene present between these two stop codons, it's not going to bridge any further out from there. ggcaller then looks within the stop codons for potential start codons. And the issue with many bacterial prediction and gene prediction tools are sometimes they predict the wrong start codon because a methionine, which is encoded by a start codon, can encode the start of a gene, but it can also encode just a methionine that's present somewhere within a gene sequence. And so if we were to call a start codon this start codon here in the middle, this may actually be the wrong start position for a given gene. So to improve this process, we use the population frequency information provided by the pangenome graph itself. So we can actually get a count of how many genomes each of these nodes is found in. So this genome is found, this start codon is found in 3 genomes, this start codon is found in only 1 genome. And the motivation behind this is that we assumed that gene truncation is highly selected against. So therefore, we would assume that if a start codon is found in multiple genomes, selection is favoring its maintenance because it's found in so many genomes. And so we favor start codons that are found more often in a population than others. So we assign this as the correct start codon, and then we can then traverse outwardly from this start codon to call all of the genes that are in all of the genomes downstream of this start codon. And this process is repeated, steps 3 and 4 repeated for every single node in the graph. So the upshot of doing this in a pangenome graph rather than in individual genomes is to improve gene prediction consistency across related genes and also to aid in clustering. So for example, if we called a set of actually related genes in 3 different genomes, in genomes individually, we may well call the wrong start codon at each of these different genes. And when it comes to clustering, we won't actually put these genes in the same cluster and may underestimate the number of core genes, for example, that we have. Whereas using correction, we ensure that we pick the same start codon for each of the genes in the population, for each of the related genes in the population, and this greatly improves our ability to cluster. I said I was going to mention Panaroo again. So at the end of this process, once ggcaller has called genes in a Bifrost graph, it then uses Panaroo, which uses, rather than a compacted De Bruijn graph, Panaroo uses a gene graph to correct for prediction. So for example, Panaroo is able to remove prediction errors, so erroneous errors that aren't supported anywhere else in the population. As I mentioned before, can be used to refine genes based on the presence of a gene in other genomes. And also, ggcaller uses or directly annotates clusters. So with functional information, so saying what the function of a gene actually is, rather than just spitting out the sequence of the gene. And by annotating in this way, it ensures that all genes in a given cluster are annotated consistently again. So we don't have 2 related genes being functionally annotated as different things. So just to show some results now, what I'm showing here is how much ggcaller improves gene prediction consistency. So in this graph, what I'm showing here is analysis of a Neisseria gonococcus dataset, and with increasing numbers of genomes in each run of ggcaller. And I'm comparing ggcaller in red against 3 other tools or workflows. So Proca, which is a linear genome gene prediction tool for bacteria, and then 3 different methods of conducting clustering. So Panaroo, which is also implemented in ggcaller, PPAN, which is another state-of-the-art gene clustering workflow, and then Rory, which Neil may have mentioned last week or previously in the previous webinar. What I'm showing on the y-axis is the within cluster of orthologous genes length coefficient of variation. What does that mean? The lower this is, the more consistent the lengths are of genes in a given cluster. So if it's lower, it means that genes are by and large the same length within a cluster. If it's higher, it means there's greater variability in genes in that cluster. As you can see, it's ggcaller is outperforming both Rory in black and Panaroo in grey when you actually increase to many thousands of isolates that or genomes that you're analyzing. Best performing is PPAN, and we're a bit stumped by this, why this could be. But if you actually look at the number of genes within these clusters, PPAN actually results in many clusters being split up. So it actually results in more consistent clusters because it splits up clusters more often. What ggcaller is doing, it's maintaining gene length consistency, so improving gene length consistency whilst maintaining large clusters. So you can see it's sort of second best in terms of gene length, but it's also second best in terms of the number of genes within each cluster. So it maintains in large clusters which are more consistently annotated. And this is because it's using this information from the pangenome graph to improve gene predictions. Also looking at computational efficiency. So what I'm showing here is the number of times faster ggcaller is when run on different data sets, so some Streptococcus pneumoniae data sets and the same Neisseria gonococcus data set, versus a linear genome workflow of Proctor and Panaroo. And you can see that in almost all cases, ggcaller provides a multiple times speed up against Proctor and Panaroo because of the speed gains you get by annotating genes once rather than multiple times across a population. Another motivation behind using a pangenome graph in this way is that we can actually make gene prediction more robust to assembly errors. So in this case, I'm looking at fragmentation, where parts of an assembly might be missing simply because there weren't enough reads, for example, to make an accurate assembly in a particular location. So gene spanning a contig break, where you have this fragmentation, can be recalled using sequences from other genomes. So I'll just give a cartoon example of that. If we have 3 different related genes across 3 different genomes, say in the blue genome and the green genome, the genes are predicted fine, there's no contig breaks. But in this yellow genome, there's some fragmentation, which means we actually can't predict this gene. They sort of would start and then stop before we actually read through the whole length of the gene. Using a pangenome graph, we can actually use the graph's structure to traverse this contig break using information provided by the blue and the green genomes and allow full recall of this gene. So what I'm showing here is a comparison of ggcaller in red with 2 different prediction tools, so Proctor, which we mentioned before, and GeneMark-S, which is another gene prediction tool. In the original assemblies of some bacterial genomes, and then ones which I've artificially fragmented. So these are the genomes that are fully intact. These are the genomes that are fragmented. The further towards this top right corner, the better. So higher recall, higher precision. What we can see here is ggcaller is quite is maybe just underperforming versus GeneMark-S and Proctor 2 when the genomes are fully intact. But if we actually fragment the genomes, Proctor and GeneMark-S can't call any genes, whereas ggcaller can call some. So this means that ggcaller actually using a pangenome graph, as ggcaller is, makes gene prediction more robust to fragmentation of assemblies. And one of the main motivations, just to move on to the sort of another result slide, one of the main motivations behind using pangenomes is to avoid reference bias. And ggcaller also aids in reducing bias caused by using single references in downstream analysis pipelines. So in the ggcaller paper, I look at the ability to annotate significantly associated variants, which I've conducted a GWAS against the given phenotype with. So for example, if we identify a particular variant that as being associated with, say, drug resistance, we don't know, we get out from our GWAS tool the actual variant that's been found and associated, but we don't know what it actually does, what is its function. So normally the process is to take that variant and align it to a reference sequence which we have good functional annotation for. And if that variant is found or aligns well to our reference sequence, we'll get the correct functional annotation. If that variant isn't found in our reference sequence, we'll align it and it may well mismap to somewhere completely at random or wrong, and the functional annotation we get out is actually meaningless. And this is what I'm showing here. So what I did was I conducted a GWAS with a set of Streptococcus pneumoniae genomes, looking at the association between different genomes with two antibiotic resistance to two antibiotics, tetracycline and erythromycin. Now, you have to take my word for it. This is the correct locus. So looking at the alignment of different variants association, this is a Manhattan plot, by the way, aligning to a given reference sequence. The tetracycline alignment of these variants is correct. So this peak identifies the correct locus, which is responsible for tetracycline resistance in Streptococcus pneumoniae. And this is because this variant is also found in the reference sequence. When we align the statistically significant variants associated with erythromycin, this gene, the particular variant, is not present in our reference sequence. We get two false positive peaks, which are both incorrect. And so this boils down to reference bias, and we need to use more diverse reference sequences in order to correctly annotate these significant variants. Using a pangenome graph, as is ggCaller, and using the annotations on that graph, what we can do is, no matter what the variant is, because we have a better representation of all of the pangenomic diversity in a population, we can annotate the, say, blue variant correctly and the red variant correctly because they're all both well represented in our graph. And what I'm showing here is just mappings of these variants to a pangenome graph annotated by ggCaller and the best and functional annotation hit produced by ggCaller. For in figure B, this is the mapping of the tetracycline variants, and what we get is a very strong signal, a map to the TET-M gene, which is the causal gene for tetracycline resistance. So matching up with our reference-based approach above. But what we see is if we map the erythromycin resistant variants to the ggCaller graph, we get hits to MEF-E and MEL. These are efflux pumps which are have been functionally associated with erythromycin resistance. So using ggCaller, we can pull out the correct functional annotations for our statistically significant variants identified by GWAS. So just to wrap up then, why would you use pangenome graphs for gene annotation? Using pangenome graphs provides consistency because you're using this population level information to inform gene prediction. Gene lengths are more similar between each cluster. You get gains in efficiency because you're not conducting this redundant annotation of genes in every single genome. You're annotating genes across a pangenome all at once. Robustness, so using a pangenome graph allows you to use information from other genomes to improve the accuracy, particularly in fragmented assemblies. And you can also use graph-based annotations to give an unbiased, provide an unbiased way of accurately annotating variants used in GWAS, for example. So just to sum up then, pangenome graphs are a compact representation of population-wide diversity, and there are a number of different methods available, and your choice should depend on your dataset, how big the dataset is, but also how diverse your dataset is. And as I've shown in the case study with ggCaller, pangenome graphs can be used to improve upon conventional bioinformatics processes by improving computational efficiency because of reduced redundancy and the compactness of pangenome graphs, improving consistency of annotation, for example, using shared information across a pangenome to inform gene predictions, as I showed here, reducing bias reduction because you're representing a comprehensive, you're providing a comprehensive representation of all the variants in the population rather than aligning to just a single reference, and also in terms of robustness, so avoiding issues with fragmentation, for example, using ggCaller and using being able to use population level information to improve annotations that way based on the frequency of a given variant in the population. So I'd just like to wrap up and say thank you to everyone involved. So John Lees, who's my PI, as well as Nick Croucher and Jerry Tonkin-Hill, who are involved in the ggCaller project. I'm just providing my blue sky and email as well. if you're interested in getting in contact, I'd just like to plug some of the tools. So Panaru, I would say the state-of-the-art tool for conducting gene level bacterial pangenome analysis. Bifrost, which is an excellent compacted drawing graph caller with a great API, and ggcrawler, which I've talked a lot about. I also wanted to shamelessly plug my PhD thesis. The introduction to my PhD thesis goes into a lot more detail on different types of pangenome graphs and some of their applications as well in looking at pangenome evolution and genomic epidemiology. So if you're more interested in reading more, I'd point you in the direction of that. There's some nice citations in there that can better inform you around pangenome graphs as well. So yeah, thank you. And any questions, I'm happy to answer. I'll just put it back to this slide so people can jot down things.
Yeah, thank you so much, Sawar, for this very clear and structured talk. So all the links will be in the slide that will also be provided tomorrow. So don't worry, you can access them. And thank you so much for sharing also the PhD thesis. Sometimes it's actually easier than reading the paper as well. So we already have 20 questions on the Q&A. So I ordered them by most voted ones. So we directly start with the first one. So we try to go over as much of them as we can. So the first is it possible to use cactus with multiple species where only draft genomes are available? More generally, if we don't have chromosome-level reference genome, is it okay to use pangenomic tools?
Yeah, it's a great question. It's always better to have chromosome level assemblies. I've definitely built graphs before, even with cactus with non-chromosome level assemblies. And I believe in the original cactus paper they were building these graphs with non-telomere to telomere assemblies. This is back in 2011, or the whole genome alignment may have been in 2017, but they definitely didn't have telomere to telomere assemblies for all of the different mammalian species that we're conducting this alignment with. So it's definitely okay to do, and the authors of Cactus have definitely done it in this specific this example of using Cactus. And yeah, of course it's always better if you have a more complete assembly to conduct pangenomics on complete assemblies, but they are relatively rare compared to draft level assemblies with short reads. So I would definitely say that you can use these tools for on more fragmented assemblies. And as I showed, pangenome graphs are quite robust when you're, for example, doing gene annotation on them. They are robust to fragmentation and incompleteness.
Thank you. Oh, sorry, the question's moved. Okay, the next question is referred to Panaru. So Panaru builds a pangenome graph but has paralogs purged. How can we resolve these graphs to study recombination hotspots and annotate the merged paralog nodes? Are there tools for such graph annotations?
Okay, so identifying recombination. Not that I'm aware of that are available publicly yet. It's definitely of interest, particularly I know that some of the Panaru authors are looking at this. So I think it's more of a watch this space type of thing to trying to identify where recombinations are. I mean, there are the Panaru software comes with the ability to identify structural variants and look for bubbles in your pangenome graph. So this may well be one approach to identify. I know that one issue with using Panaru is the ability to identify mobile genes, and the issue being that these mobile genes, they should be sort of merged as orthologs across different genomes, but tend to be split as paralogs because they move around all over the place, and Panaru uses synteny to conduct a lot of its QC measures, and so mobile genes do tend to be split up, and it's more difficult to conduct analysis of mobile genetic elements using something like Panaru or any synteny-based approach because this synteny assumption breaks down.
Thank you. The following one is on OrthoFinder. So someone is
Okay.
asking if it's a useful tool for identifying presence, absence, variation genes to categorize core, accessory, and unique genes with a pangenome.
Yeah, so AutoFinder, if I remember correctly, does not use a pangenome graph. I believe it uses sort of reciprocal best hit blast searching. So using um, sequence identity to look for genes that are similar and placing them all in the same cluster, and then identifying paralogs based on sort of whether or not two related genes in one genome are more similarly related to one gene in a different genome or another gene, and splitting paralogs that way rather than using synteny. I think that AutoFinder is useful for when you're doing genome comparisons between multiple species, because this idea of if you're conducting sort of a pangenome analysis of a genus or higher than a genus, this the assumption of shared synteny breaks down between your genomes. So Panna is really good for analyzing single species data, whereas something like AutoFinder would probably perform better if you're doing comparisons between multiple species.
Thank you. Then another attendee is asking, is there any recommended size of a variant to call as a bubble in the graph?
I think it depends on the graph method you're using and whether the graph approach can actually identify a bubble. If you're using the Breuil graph approach, it may be a lot more difficult to sort of tailor which size variant you're interested in, just because of the way that k matching works. You may well get sort of messier graphs where it's more difficult to identify what truly is a bubble versus what is just random k matching. Um, Using something like a variation graph. Variation graphs can be built from a number of different tools, so you can build them from cactus graphs. Variation graph also has its own implementation of methods for building graphs and conducting alignment. I wouldn't say there is sort of a maximum size. I think it just depends on the graph building tool that you're using and whether the graph building tool is able to actually represent structure variants of that size, and whether you're confident that those are yeah, it's capable of doing that.
Thank you. I will then take a question on the Bruin graphs because you mentioned it now. I think it can be related. So someone is asking, when building the Bruin graphs, overlapping k-mers increase number of nodes. Why can't we use non-overlapping k-mers and edges by their construction, then determine the next few bases in the genome?
Okay, no, non-overlapping k-mers. I see. So the issue you have is if you're using non-overlapping k-mers, then you have to sort of think about where in the position of each genome that you're looking at and starting from, and the stride that you use. If you don't use non-overlapping k-mers, you may well call being a certain window of a genome, well, in one window starting from one specific location in one genome, and calling all of the non-overlapping k-mers moving forwards. In a different genome, you may well be starting from a completely different locus when you're calling the first k-mer, and you jump by a specific stride, which means that you may well, if you're sort of one or two bases off or like a window size off, you won't get any k-mer matches between those genomes. So using overlapping k-mers means that you're guaranteed, it doesn't matter where you start in each sequence, you're always going to get matches because you're covering every single sort of window along a genome rather than jumping between them. I hope that makes sense.
Yeah, thank you. Next one is on monomorphic organisms such as Mycobacterium. Will you advise using alignment-free graphs for this kind of organism?
I think so. Yeah, I guess so. They're super undiverse. Something like Mycobacterium tuberculosis. Yeah, I would definitely recommend using something like a alignment-free approach because they're so undiverse. You won't run into issues of multiple k-mers mismatching because two variants appearing in the same k-mer, for example. So you're going to get very compact graphs with very few bubbles. So I think it's probably one of the best possible applications for these kind of graphs. Yeah.
Thank you. Next one is, How can gene-centric pangenome graphs be adapted for genomes where diversity is primarily intragenic and driven by recombination rather than gene presence or absence?
Yeah, so I guess the main reason why gene graphs are so popular in bacteria is because we have very good annotation tools for genes in bacteria, and gene centric well, bacterial genomes are gene rich. So looking at the genes, you're covering most of the genome. Anyway. But there's nothing limited, sort of stopping you from representing each of the nodes in the Pangene in the Panaru example that I showed. They don't necessarily have to be genes. They could be any kind of functional annotation possible. Then it comes down to an issue of how do you then cluster sequences that aren't genic, that aren't necessarily under strict or as strict functional constraints or evolutionary constraints as genes. We have very good gene prediction tools and gene clustering tools that work on amino acid sequences. It becomes a bit more difficult to then start clustering intergenic sequences where there is no translation and there aren't all these tricks you can do to cluster amino acid sequences. So there's nothing stopping you from sort of annotating these gene-centric graphs as sort of more functional agnostic graphs where you can represent both genes and intergenic sequences. It just becomes a bit more tricky in terms of how can you then determine what's orthologous between two genomes if you're not just looking at genes.
Thank you. Next one is, are these tools directly applicable to amino acid sequences?
Um, well, it depends which graph-based tool you're using. A lot of these tools use tricks for sort of minimizing memory consumption by encoding DNA bases as specific floating or specific uh, 8-bit integers. So some of them won't specifically be useful for amino acids because of this issue, but there are definitely tools out there that you can build that allow you to build the brain graphs from amino acid sequences rather than nucleotide sequences for sure. And all of the sort of the benefits of using a pangenome graph in for genomes also would apply for using them on amino acid sequences as well.
Thanks. The next one is, which type of graph amongst the one you mentioned is appropriate for most diverse population?
Yeah, I think probably mini-graph cactus. Sorry, cactus. There's an implementation of mini-graph and cactus where it's combined as well, which I've used and seems to work quite well. I'd say that is probably the state-of-the-art tool for doing very diverse, very distinct alignments across. It's really designed for conducting whole genome alignment across whole sort of eukaryotic species. So it's probably the most flexible out there at the moment. Yeah.
Thanks. Because there's a question that is related because someone else is asking, which type of graph can be used for bacterial species that are highly prone to mutation?
hmm Yeah, I mean, I've used alignment-free graphs. So Bifrost is really good for bacterial genome analysis. If your population is too diverse and you're seeing sort of your graphs are too disjointed where you're not getting a lot of k-mer matching, you can reduce the size of the k-mer. So by default, Bifrost uses k-mer length of 31, which is quite long. You can reduce that down and it will mean that you're less sensitive to sort of super like hypermutators where you may well get a lot of mismatching k-mers. So reducing k-mer size is a way of being able to apply alignment-free tools to more diverse species.
Thanks. Next one is also regarding eukaryotic. So someone is asking, what is the best approach to align a mini graph-based genome graph with gene annotations in an eukaryotic haploid genome, or is it better to align only the target gene for downstream analysis?
So, sorry, could you repeat that?
Yeah,
What is the best
that's
approach?
it.
Okay, I
Yeah,
can see it here.
I will repeat it.
What? Okay, yep.
So the question is, what is the best approach to align a mini graph-based genome graph with gene annotations in an eukaryotic haploid genome, or is it better to align only the target gene for downstream analysis?
Yeah, I'm not aware of approaches that can be used for annotating mini graphs in specifically. Maybe there are new approaches that I haven't seen quite yet. Um, yeah, I think it's probably better if you're sure of the gene that you're interested in that has mutations. Um, Minigraph will only be able to represent structural level variation. If you're interested in how, say, a gene is moving about a genome, I would suggest, yeah, building the minigraph graph and then aligning your gene of interest to see sort of where it pops up to that mini graph. And there are ways of doing that in the VG toolkit to align a sequence to a pangenome graph. That would be probably the best way to do that, in my opinion.
Thank you. It's almost time. We have 1 minute left, so maybe we can take a couple of questions more before we close the webinar. So the next question is, the nodes in the Pangenome graph by PGGB have nodes of variable length, smallest being one base pair. Are these graphs used in graph convolution networks, deep learning approaches?
Mm-hmm. Yes, it's a great question. I'm not really. I've only seen one application of pangenome graphs in deep learning approaches so far that I've seen that's convincing, and it uses minigraph. And the reason it uses minigraph is that it can order the nodes in a particular way relevant to a reference, and so can use sort of transformer-based approaches like used in ChatGPT, for example. Um, it's something that I'm still thinking about. How can we use pangenome graphs in sort of a deep learning approach? It's not the sort of the application to me isn't straightforward. There's not really a quick and easy thing to say, okay, this is definitely going to work, and this is the application for deep learning and when applied to pangenome graphs, because graph convolutional networks are mainly used for node labeling and are trained on, you need to train them on sort of stable graphs that such as protein-protein interaction networks, which don't change. They are sort of known to function in a specific way. Pangenome graphs vary depending on the genomes that you're using to build them. And so it's very difficult to say, okay, what do we want to be at? What's the actual reasoning behind training something on a pangenome graph? What do we actually want to label these nodes? How do we train a convolutional network, for example, on the pangenome graph when we can only train it on one specific graph, and when we build a different graph, the structure is going to be completely different and will almost certainly not generalize. So it's quite a difficult question and trying to work out. It's definitely something I'm thinking about. How can we actually use and train deep learning approaches on pangenome graphs, and what are we actually can we actually do with that? What is the research question that we're trying to answer?
Thank you. I will now take a more practical question that came in while you were talking about the brain graphs. So someone's asking, how does this methodology scale with the number of genomes to be analyzed? I'm referring to hardware usage because visualizing graphs can be computationally intensive depending on the size.
Mm-hmm. Yeah, I think visualization is a tough one. Once you get to a few thousands of genomes in a graph, they particularly with the Breuil graphs where they can become quite messy, visualization often isn't super helpful. In terms, I think visualization of more than a few hundred genomes is not really that useful unless there's ways in which the VG toolkit tries to simplify visualizations, which is very useful, and sort of tries to linearize pangenome graphs, which is only really possible in sort of less diverse species. Um, Saying that, if you're not looking to visualize pangenome graphs and just sort of write tools to analyze them, to align to, some of these tools will scale to hundreds of thousands of genomes. So Bifrost, in the Bifrost paper, I think they run it on 100,000 bacterial genomes at once, and it scales to that level, and you can conduct analysis and read in that graph. And there's also kinds of sort of memory tricks that they use to improve the ability to load that into memory and manipulate the graph in that way. So it depends. Visualization depends how many genomes you've got, and probably it's not going to scale beyond a few hundred genomes, and depending on the diversity of your pangenome graph. But if you're looking to not visualize your genome and conduct downstream analysis, you can run these built graphs from hundreds of thousands of genomes.
Thank you. I think we have time for one last question. Someone is asking, are there tools available for interactively exploring pangenome graphs similar to genome viewers? So I believe like user-friendly, let's say.
Yes. So Bandage is a very robust one. It's been around for quite a while, but is sort of super useful for looking at structures of your graphs. You can annotate the different labels aligned to your graphs, look at say presence or absence of certain genes in your graphs. Bandage is a really good one. I've seen examples of more recently examples of De Bruijn graph, or just in general pangenome graph visualization tools that are out there. I'll have to. I believe there's a new one called Pangenome XG, which I need to have a look at. There are a number of different tools available that you can use to sort of have a look at. I have used Bandage before, which is super useful for just loading in and looking at the structure of your graph. That's the one that I would use.